Okay, so our next speaker is someone also very special, and you will now get to know her in the FAST community. She's going to be a newbie for today, but hopefully very quickly you'll welcome her with open arms. So those of you that know me and Paula know that we always find people that are incredibly valuable and talented as quickly as possible and get them to join our cause, because we're passionate moms who will do whatever we need to do to help our children and yours. And so this is a nice example of that. Just to give you a little background about Lauren Black, Dr. Yu, who we completely manipulated to join the Angelman cause very quickly, and he agreed, we were talking about our program and our foundation and our mission, and he really led us to somebody who he had the most high regard for, and who was very instrumental in really helping Mila's program be successful. And he talked about her multiple times in his talk, and I felt very strongly that if she's good enough for you, she's good enough for us. So I felt that if she was willing to join our cause as well, then we would be very lucky to have her. And so I sent her an email about me and my daughter, and I had a response in 45 minutes and a phone call that lasted an hour and a half, about an hour later. And she was all in. She jumped in that night. And she has been probably one of the people that I talked to the most in the last 4 months. But she has really been instrumental in bringing some of our programs forward. So I want to welcome her. Dr. Lauren Black is a distinguished scientist at Charles River Laboratories, but most importantly, she'll tell you a bit about herself and her history, so I don't want to steal her thunder, but she really is a true liaison between her past life. She's like a failed FDA retiree almost, her past life at the FDA and her future life in industry and drug discovery, and she was able to see both sides of that coin, and she really has an unbelievable ability to relate what the FDA needs and wants and why that's so important, and to help all of us realize that they're our friend. They have regulations for a very important reason, and that is the safety of our children. And how can we use that and understand that in order to ensure that we're choosing the best drug candidates for our children in the safest possible way? And so she has really helped to lead our mission going forward in drug discovery. So I'm very excited to welcome you to speak to our group. And if you would come up, that would be wonderful. A warm welcome. Mode, okay? Like this? You can advance either of the arrows, and laser doesn't work. So if you want to point to anything, just use the mouse. Okay, great.
Well, I haven't talked in front of such a big audience in a long time, so I'm trying not to be scared, but I see a lot of really friendly faces out there. So just smile and wave once in a while, and I'll, relax. But I am just so excited to be here. I'll tell you a little bit about my history, but I wanted to say that I'm very grateful to have the opportunity to bring some of my past history to bear on your clinical challenges and your children and the families' lives that are represented in all of the people and the faces here. All of our history contributes to all of our future. And what we're going to do is make cures happen for these children and many other children with rare diseases, because the technology has come to the place where we can make that happen. And I believe deeply that there are benefits on the horizon for all of the families here in one way or another. And I especially take these notes about getting on the registry. I have a friend who has 2 children that are going blind, and the family is very reticent about trying to get their children sequenced. And I told them, I said, if your 2 kids have a mutation that would be something that was shared by a few other people, well, maybe a drug will be developed for your children. You have to get registered in order to make that plain to the people who can fund those new projects. So I wanted to tell you a little bit about my personal journey. I'll try to keep that brief, but it also goes to show that each person's history becomes part of the future of many people. So that's why just take it as a point that each of our histories and all of our experiences in our family make it possible for us to contribute to society in many different ways. I actually went to internet and pulled the exact same figure that Allison did, only I didn't know how to cut and paste it in properly, so I took my cell phone and took a screenshot and then sent it as an email to myself. You got it. What a clueless, anyway, so it's fuzzy, right? All right, and I just think it was so funny that we went to the same picture. If anybody wants to read up on details of drug development, this came from a really nice publication that's available as a brochure from the organization PhRMA, PhRMA, and if you go to their website, that entire brochure talks about all the different parts and stages of drug development. But please bear in mind when you look at that, you'll notice in the fine print at the bottom, that is fuzzy because it's a cell phone screenshot, they talk about thousands of patients. Well, no. That's not really applicable to your case. When we do these patient studies in in rare diseases, we can often get drug approvals with a very small patient database. It all comes down to the outcome measures and whether you have sensitive markers, just as we heard from Dr. Reeves. So if you can get good outcomes and be able to detect the changes, you can often get what's called a conditional approval from the FDA with that kind of data, because there are no other therapies out there, right? So you're being held to a type of a novel criteria when you're the first person that can help address the issue. Well, you're going to get a better treatment from FDA over that. So what our job is let's see if I can make this pointer work.
It's not where you want to be on this slide here. You want to be on the next one there. So that's the one you were just talking about. So you see it forward.
There we go.
And then use this as your pointer because you can see that.
Thank you.
Okay.
The IT complications, right? So what we, yeah, that's right. You got a big briefer on it. So basically, the other thing I wanted to point out about this, I can't get it to work either. Never mind. Just to say that, remember that looks like a really steep funnel. It starts here and it gets really skinny. And what we want to do is we want to use your funds really carefully, right? So we don't have the money, all of us, right? $50, $100 at a time over many thousands of people. We don't have the money to start off 1 billion drugs and come down to 10, right? We want to start off with 100 drugs and winnow it down quickly to 10 and make sure a couple of those work. So we want that funnel to be kind of narrower from the beginning and then make sure all along the way we use the best science to try to make sure that we've selected the right drugs, keep them in the pipeline, see if they work, see if they work, see if they work. Not all of them will work, right? And then when we get to the end of the day, we have several drugs in our pocket that we can work with. It'll be used for very different kinds of things for the kids. Some of them will help their quality of life, some of them might help the EEGs, et cetera. And some of them might actually reverse the gene problem. So what we're going to be doing is trying, Allison referred to my work in drug discovery, is we're going to try to make sure that the selection criteria for those drugs is rather narrow so that we don't have to fund a whole bunch of drug programs in order to get one candidate. Does that make sense? We want to start fewer and make sure they're very successful and a higher proportion of them are successful. Now, going back a little bit to my personal journey, when I really want to scare my 2 children, maybe I should make a point here. I used to have 3 children, right? Well, I started off with 1, and then I had another. That's the usual way it usually works. The second child, unfortunately, was probably, and Dr. Yu told me this, probably subject to a, what's the right word, mosaic mutation. Unfortunately, he had he was a perfect boy, but he had a severe mitral valve problem. We talked about that last night. So everything was perfect except for this one little piece and part. This was 18 years ago, so I can kind of talk about it now. But basically, I spent 3 months trying to save him. The doctors at Children's Hospital of Philadelphia and Fairfax, Sonoma attempted a couple of really major surgeries, but it didn't work. And I think that what I did learn from that is what it was like to live in a parent community. I was at Children's Hospital Philadelphia's Ronald McDonald House for 2.5 months while my child was in the cardiac intensive care unit. And we watched some miracles occur. 75% of the kids in that unit pulled through with amazing results. Unfortunately, we were not one of that team. But I subsequently had another child who was healthy, and I feel extraordinarily blessed. But that's part of my journey. And today, 18 years later, I'm finally through work like with Tim last year and with Allison and her team this year. I'm finally able to embrace both sides of my history, both at FDA and as a mother. And I can talk to you without crying, and I can work on this field of rare and orphan disease and bring everything that I've learned from being a parent and from FDA days and from work at NIH to bear on this issue without feeling that sense of depression or loss of hope. So I want to tell you that in working through this, my sense of hope in my personal journey has become a commitment to help people like yourselves. I spent, thank you. I've never told that story in public before. So, okay. If I really want to scare my existing 2 children, who are 19 in college and 15 in high school, I tell them I spent 23 years in school, I guess because I had to repeat some classes. And then I worked at 2 years at NIH working on cultured neuronal cells. So I was actually a neurobiologist for a couple of years looking at dopamine signaling. And after that, after finding out how hard it was to be a bench researcher, oh my God, I decided I was going to get a desk job. And I thought, oh, I'm sort of retiring early, I'm 31. I'm just going to go sit down and have a nice science job and take it easy. And that's not what happened. I went to work for the Antivirals group at FDA during the AIDS crisis. I didn't really know that that's what was going on and what I was getting into. I spent my time there learning about rats and whatever else we use in drug development, and learned that really animal studies were very difficult. I had to learn a lot about animals. I'm not a veterinarian. And meantime, while we were studying and trying to work on these 1-month decisions for INDs, AIDS patients were dying. They were dying of fulminant pneumonia. They had nothing to really eradicate their virus, and they were protesting in the streets. And they had every right to. We learned so much today. If anybody wants to know how to do patient advocacy, go back to the stories of ACT UP during the early 90s. They were amazing. And a lot of the things that you're doing sort of are fueled by the work that was laid out by not only the advocates for AIDS, but also subsequently the Komen group for breast cancer. And then the patients started to take power, and the FDA started to listen to the patients. That started back then, and this is what you're capitulating now when you bring the Angelman kids, syndrome kids into the agency and introduce them. That door wasn't open until the AIDS patients opened it. So finally, FDA started offering compassionate use, conditional approvals, based on viral load decreases instead of complete cure. And then the first types of pre-IND advice in which the FDA actually took put on a consulting hat. They were the ones who, the antivirals group, our group, was the one who started that. And then that's become part of the law called PDUFA, that all sponsors have the opportunity to come into FDA and get some advice before their INDs, before their clinical trials start. At the time I was there, the protease inhibitor, the HIV protease that's so important for the replication of HIV was published. And during the time I was in the antivirals group, just a few years later, they were able to approve the very first protease inhibitor that's been the keystone of almost curing AIDS. People live for very long now. So I watched this miracle occur in front of my very eyes, going from protests in the streets and death to being able to see a drug come to fruition within just a few years. That's how I cut my drug development teeth, and that's the place I've stayed. So at the same time, I got assigned, I raised my hand for a lot of weird drugs, And I got assigned the first oligonucleotides for HIV. Unfortunately, the first generation drugs caused all or nothing kind of shock reactions within just a 3-fold range of concentrations in the blood. Now, remember, I was trained as an in vitro scientist with cells, and so, you know, I'd been really well educated about the importance of concentration response in vitro. Well, so it wasn't like a big leap to say, well, we really need to understand the concentration response. Let's just look at the blood volume of our animal and say, okay, that's like a petri dish. It's the concentration in the blood that matters. Well, in fact, I did some analytical work, and indeed, the concentration in the blood was exactly what was causing the problem. So we were able to put in monitoring on those clinical trials that were very, very careful monitoring for blood levels and go ahead with those studies, even though these drugs were very toxic. Subsequently, as you heard from Tim, and as you'll hear from Scott and others, there have been novel second and third generation oligo drugs that are more safe. So, but as you see, it took the first generation to get started. We learned a problem, we applied new solutions to it, and then we've learned more and applied that and developed safer drugs. That's the way drug development always goes, and you're at the beginning of that pathway too. But you're taking advantage of that knowledge that came out in the early 90s. So you don't have to repeat, what did Dr. Reeves say? You don't have to reinvent the wheel. So in working with both tacrolimus for liver transplant and these oligonucleotides that had very narrow safety margins, that's a term that I think you should all memorize, take home and write over 5 times. Safety margin. All drugs are toxic, but what is the safety margin? You know, even if you take two rounds of Sudafed, do you know what happens to you? Oh my God, don't do that, right? One time I did, you know, I ended up having, you know, practically passing out because it's actually has a very narrow safety margin, but it's not over the counter. So you have to be cautious with these agents just like you do Sudafed. Having run out of difficult issues at Center for Drugs, I decided to raise my hand to go to the frontier, which was Center for Biologics. I worked there another 7 years. At the time I was there, things got even weirder. We started to look at porcine cell. That's pig cells, like fetal pig cells injected into the brain. They were doing this in humans. And it looked like it was going to be a great thing until we found out about something called the Porcine Endogenous Retrovirus. Well, okay, it turns out that there's a virus in pigs that we don't want to get in our brain. So that kind of put a kibosh on that for a while. And it's coming back now because they've actually CRISPRed pigs to get rid of their retrovirus. I don't know if you've heard about that. And then Adenovirus Therapy started up. And then that was the first series of vectors, and now we've improved on that, and I think we're studying more AAV vectors, which is a different type of virus. At the time this was going on, they were also, for instance, Duke, I think it's Kurtzbaum? Judith. Anyway, I can't remember her last name. She was doing a fetal bone marrow transplant of the father's bone marrow into the growing fetus inside the mother. And they knew from older children that the older children had severe combined immunodeficiency, and they started the protocols where they take the healthy father's bone marrow and inject it into the growing fetus of the second child of the family so that they could save that child. And that started to work. I've seen so many miracles. I mean, it's just been amazing. T cells were being turned into tumor fighters at NIH, and we know today that we have CAR T cell therapies that are curing children of cancer. Artificial pancreas and artificial liver devices took cells from cadaveric liver, dead people's livers. They would chop them up, stretch out the cells, and line capillary tubes, and then do sort of like a heart-lung device. They would make the tubes, take the patient's blood, run it through the tubes, and then the cells from the cadaveric liver would digest the intoxicants. And this would replace, like someone, if their liver was failing, this would life support for them and help digest all of the nasties in the blood so that they could wait for their liver transplant and still be alive. I hope that makes sense. Anyway, so you had a cell device combination that could bridge someone to transplant. They also took macerated or cut up pancreases and they're able to make islets out of them and infuse them into the liver. And they took brittle, life-threatened diabetic patients, injected the cells into the liver, and were able to completely get these very, very, very bad diabetic patients to be completely off of insulin. That works sometimes. So in the course of this, I was reviewing 2 or 3 INDs a week for these kinds of high-risk files. We reviewed these very lean toxicology programs that were designed by academics in most part, not necessarily run in big CROs like I work for now. And I learned the very delicate balance between the importance of informed consent, to not raise too many hopes, to make sure we selected the patients who didn't have other therapies for these very high-risk trials, to make sure the parents and the families really and the patients really understood the risks, because the animal studies, as you heard this morning, are not perfect in any stretch of the imagination. We have to do them. We learn some things. We can't learn it all. And I learned that as an FDA reviewer, when I was doing the sign-off on this, it was a nerve-wracking experience because I knew I didn't know everything. And I was trained as a researcher. I wanted to know everything. I wanted to do a doctoral thesis on every single IND, and I had 3 days. But when I took that pen to paper, I had to make sure that I felt comfortable knowing that the patients in this trial were just like my kid or my mom. And everyone at FDA that worked on those files felt the exact same way. So when you go down and talk to FDA, you're talking to mothers and brothers and children. And they look at their jobs as public servants as protecting their own family. And I think what you need to do is make sure that you talk your family to their family and express it that way. And I think you guys are really doing a great job of that. How much time do I have left? 3 seconds? Okay. Oh, I can't do that. You've got a lot of other things to do today. Okay. So to form this into what you can take forward, I'm going to try to move really fast now. We had setbacks. What are setbacks? Okay, that's a nice word for saying some patients die on trials. Some patients have severe adverse events. Remember, these are I'm talking about extremely sick patients in the most part for the trials that I worked on at CBER. But it was these were rare events in most, I would say 99%, or maybe 999 out of 1,000 cases. We had, I would say, extraordinary safety and really good progress. Because we don't have forever. Remember, the diseases are progressing. The diseases are overcoming and killing patients. You have to look at the opportunity to cure someone in the context of their disease is going to kill them. So it's, you're playing off checks and balances on, is it possible that this therapy will help them because there aren't other choices? We're not talking about acne medication or cosmetic indications or even upset stomachs here. We're talking about very high-risk disease. So those setbacks were bound to occur because these are truly clinical investigations. Remember that the AIDS research programs of the 1990s that were a public health crisis taught us amazing things about T-cell science, immune science that was brand new. We developed all kinds of laboratory technology for helping assess T-cell responses. T-cells are the things that attack tumors, they attack viruses. We needed to really understand T-cells for the first time, and all of a sudden we had new technologies arising from basic research. But this was new science arising in the 90s and being applied in the 2000s. We learned that some immune suppressing antibodies, we understood the pathway in the culture dish, but when we put them into people, they developed shock. Well, why would an immune suppressant antibody cause an immune stimulation? Oh my God, right. Well, we had to learn a lot of patterns emerging from things and then put checks and balances into place that were taught by those patterns. We studied the first cell and gene therapy protocols, but they are very complex biologics. A cell as a drug, I mean, you can't just run it through a mass spec and say, okay, it's pure. It's not pure, it's a cell. It has millions of things in there. So what do you characterize? When you made a batch of cells and tested it in animals, well, then you made another batch of cells and tested it in people. How do you characterize that those two batches of cells are identical? Well, you can look at them under the microscope. Well, they kind of look the same. Oh, wow. Okay, that's not very good. So anyway, we gradually had to build more and more quality controls and technologies into assessing these complex products. And we found that certain of those complexities might contribute to toxicity. But those patterns needed to emerge from human data. And then we also learned that in multigenic, immunologic, or infectious diseases, remember there's a lot of stuff going on. If we're septic from an infection, all of our shock mechanisms turn on all kinds of other problems in our body. And sometimes the drug in that context, which it had never been in before, would trigger unpredicted reactions. And remember, most of our trials were in patients, not in normal volunteers. So overall, these events were very rare, and we utilized the learnings every single time we got a new drug that was even vaguely like that. We would employ all of our historical knowledge and all of our experience to make sure the same things that happened on the last trial never happened again. And now you've had the advantage of these 25 years experience, and the fact that I'm a baby boomer means that when I left FDA, a lot of people didn't. A lot of people, I had 11 years there, they've had 30. They're still there. That's kind of a good thing or a bad thing. But in any case, they have a lot of historical knowledge and are still passing along that knowledge to the new hires that are coming in today. Good news, the roads were paved these last 30 years, and you have the advantage of that. And I think that if you ever want to be inspired, not only from the Mila case and Dr. Yu's work that you heard about this morning, but take a look at the Kymriah. You can see it's Kymriah from Novartis, and you can go to the CHOP and Hospital University of Pennsylvania web pages, and you'll see some really beautiful YouTubes of a little girl named Emily, who was their first patient that was treated with the CAR T-cell, Kymriah, and she is 6 years cancer-free recently, and 14 years old and beautiful and 5'10". And it's an amazing story to watch. And the other thing, we already heard about Luxturna, the vector for blindness today, and we also heard about Spinraza from Dr. Yu. So I think that these several drugs, which represent both cell and gene therapies and oligos, are really the same kinds of drugs that you guys are considering for treating Angelman's. They're for genetic disorders, and over the last 20 years, we've learned a tremendous amount to cross-apply to the Angelman's drugs. So much has been learned that we can cross-apply. As you've heard from some of your scientists, and we'll hear later today, many of these tests are being developed in rats that can cross-apply. And even while I was hearing Dr. Anderson talk about EEGs, I wrote a note to Julie, Dr. Duville, who works in our lab, and I said, are we doing EEGs? And she says, oh, we're validating it for telemetry in next spring. So that means that we can take our toxicology animals and we can do remote recordings on them, like by Bluetooth, when they're not even aware that we're tracking on them. We're going to be able to soon do sleep studies and EEGs on animals. And the reason I'm bringing that up is when you hear that there's a patient issue, like in your patient population, epilepsy is a problem, right? What if one of your new drugs made epilepsy worse? We would want to know that, right? Right. So that's the kind of logic that we cross-apply. When we do an interview and we ask you, like, what is important to you in terms of your particular clinical indication? If I get a new case in, this is how I do this. I peel the onion and ask a lot of questions. And I ask, what is important to you? Well, if epilepsy is important in this patient population, by darn, we'll build those measures into our animal studies. So I think I should probably not take more time, but if anybody wants my slide deck, I'm more than happy to send it to you. I think I spell checked it. And I would just like to say that in the rest of my slides, I have some tips and tricks for how I classify how to approach science for a new drug. And I have a couple of pointers on the fact that there are some accelerations available for cell and gene therapy. For instance, we mentioned one this morning, Dr. the first question I took when Tim's talk. Anyway, someone
Baudet.
asked, yeah, he asked about whether or not you could use rats or monkeys. And I said, well, it has a dependency on whether or not you actually have the pharmacologic action in that animal. And that's actually our very most important question ever, is there a sensitive species that characterizes exactly what the patient's pathway can do? In that particular drug's case, the answer was neither a rat nor monkey nor dog nor rabbit nor nothing emulates the way the drug would work in a human. So the toxicology sort of takes a checkbox kind of mentality. There is a chemical toxicity that you need to risk assess for, but the pharmacology of modulating the gene wasn't going to happen in the animal. Now, in the case of some of the Angelman's oligos, we do have more species that we can look at, and the patients are healthier. So that's the kind of playoff that we get into when we start talking about regulatory acceptability and how much toxicology and safety is appropriate. For a very well patient population, we have to be extremely careful. So I'd like to close by saying I am so impressed with all of you. And I love what FAST is doing. I've learned so much more about it from talking to people last night and today. And I can't thank you enough for inviting us to be here and to work on your very important, important families. Take care now.
I think we have time maybe for one or two questions while we get Megan transferred over with her own computer. So anybody have any questions for Dr. Black? Looks like we have one back here. Any questions? Raise your hand high because I can't see you if you do. Yes, one over here. You see that, Sharon?
Oh, I was just going to mention, if anybody wants to write down my email, I'll send the slide deck. lauren.black@crl.com, as in Charles River Labs.
And we'll also have all the presentations up on YouTube that you can rewatch and look at her name and how to spell that. So that'll be in the next couple days as well. Okay, no questions? Okay, thank you so much, Lauren.